Hello guys, ayun, for today's video, we're going to uh, rationalize this science questions about macromolecules or biomolecules in mitosis and meiosis. Okay, so number one we have here, which term is the monomer of carbohydrates? When I say monomer, they are what's this, building blocks or simple forms of given macromolecules. Our macromolecules are carbohydrates, which are the sugars, and we have the lipids, where fats belong, and we have the nucleic acids, and we also have the proteins. Okay, so, lipids have the building blocks of fatty acids and glycerol, and proteins have the uh, amino acids as building blocks or monomer, and nucleic acids have nucleotides as their monomer. Okay, so ganun siya. So which term is the monomer of carbohydrates? Monomer of carbohydrates are simple sugars like glucose, galactose, and uh, maltose. No. Glucose, galactose, and fructose. I'm sorry. And that is, they are called monosaccharides. Okay, so monosaccharide is the monomer of carbohydrates. Next, number two, we have what happens to an enzyme when placed in boiling water. It becomes sugar, it is denatured, it becomes stronger, or speeds up the rate of reaction. Okay, so when you are going to put, there are two factors that affects the enzymes. Okay, so they become denatured when there is high temperature, or uh, the pH level is acidic and basic. Okay, so here it becomes, if one of them is like one factors that makes it um, denatured is the temperature. Okay, so temperature, if you're going to put the enzyme in the boiling water, it becomes denatured. And when you say denatured, is something like this: if you are going to put rubber bands in the boiling water, what happens to the rubber bands? Usually they become expanded and inelastic. Okay, that also happens to the sorry for that. This also happens to the enzymes if you're going to put them in the boiling water. So that is why when we are sick, we need to, like, so let's say we have fever, we need to just take a rest, because this high temperature or body temperature, that becomes the enzymes denatured. So that is why we become weak, because our body processes are not that in place. Okay, so that's the reason why. Okay, so denatured proteins, if we're just going to take a rest, they become we these proteins or denatured proteins can recover and becomes active again. Okay, so that's the reason why. Okay, so number three, we have choose the pair of terms that correctly completes this sentence. Nucleotides are to blank, and as blank are to blank. Okay, as I've told you in number one, nucleotides are building blocks of nucleic acids, and amino acids are building blocks of proteins. So we say nucleotides are to nucleic acids, and amino acids are to proteins. Okay, so that's letter A. Next one. Which of the following statement is true about catalysts? Okay, catalysts slow down the rate of chemical reactions. That's wrong. All enzymes are catalysts, or all catalysts are enzymes. Catalysts lower the reaction or the activation energy of the chemical reaction. That's A and D are wrong. So it's just between B and C. And remember that all enzymes are catalysts. Okay, all enzymes are catalysts. So that is letter B, because other catalysts are chemicals. Okay, so not all catalysts are chemicals. Others are, most of them are enzymes. Okay, so there you go. B is the correct answer here. All enzymes are catalysts. When you say catalyst, they speed up the chemical reaction in the body process. Okay, there you go. Next, the active site is where blank Active site is where the substrate binds with the enzyme. Okay, so there you go, the substrate. I'm sorry if you hear some chickens. I'm in the province anyway. Anyway, so there you go. Next one, number 6. Which of the following is not an example of protein? Okay, not example of proteins. Proteins, enzymes, hemoglobin, glucose, or egg whites. Okay. Enzymes, hemoglobins, and egg whites are actually made up of proteins. And glucose is not, because glucose is a type of monosaccharide, and monosaccharide is the building blocks of carbohydrates. So letter C is the correct answer here. Next one, number 7. What is the term used to describe the energy needed to get the reaction started? That is what we call activation energy, which is letter C. Next one, which of the following is not a carbohydrate? Okay, chitin, cellulose, lactase, or starch. Okay, chitin is the primary component of exoskeletons of the arthropods. Okay, cellulose, and that is made up of polysaccharides anyway. And polysaccharides is carbohydrate, so letter A is a carbohydrate. Letter B, cellulose. This is a type of polysaccharide. Okay, cellulose, and that is primary component of plant cell wall. So again, polysaccharide are carbohydrates, so that means letter B is a carbohydrate. Letter C, lactase. Lactase is an enzyme. That is working with lactose. Lactose is the disaccharide. Okay, lactase and an enzyme is an enzyme that works with the lactose disaccharide. So lactase is an enzyme, and enzyme are proteins. So this is not a carbohydrates. Okay, so letter C is the correct answer here actually. Letter D, starch. This is a polysaccharide, and this is made up of glucose, which is the sugar, or the building blocks of sugar, the glucose, and that is carbohydrates, letter D. So letter C is the correct answer, not the carbohydrates, because it's an enzyme, and enzyme are made up of proteins. There you go. Next one, number 9. Enzymes affect reactions in living cells by increasing the blank. Okay, enzymes affect reaction in living cells by increasing the rate of product formation, or the chemical reaction. Okay, so that's the letter B. Next one, a substance that accelerates the rate of chemical reaction is called a catalyst. Okay, catalysts speed up the chemical reaction. Next, we have here true or false. An enzyme increases the rate of reaction by decreasing the activation energy. That's wrong. Okay, it's not decreasing but increasing the activation energy, okay, to make it true. Next one, number 12. A product binds to an enzyme active site. It is false. Okay, so it's a substrate. Okay, a substrate binds to the enzyme's active site. Next one, substance present at the beginning of the chemical reaction are called products. No, before the chemical reaction, we call these substances as reactants. Products are the results of the chemical reactions. Okay, so this is false. They are called reactants. Number 14. DNA controls the sequence of the formation of amino acids. That is wrong, because amino acids determines the DNA. DNA formation. Amino acids, we call them codons. Okay, so codons. So it's the other way, the opposite of this. Amino acids controls the sequence and formation of DNA. Okay, so number 14 is false, because it's amino acids that controls the sequence and formation of DNA. It must be reversed. Okay, there you go. Number 15. The name of the enzyme usually ends in ASE. Most of enzymes usually ends with ASE, like protease, lactase, carbohydrates, like that. But other enzymes end with IN, like pepsin and trypsin, like that. Okay, so not all enzymes end with ASE, so that's false. Okay, there you go. Next, two substances are combined and new color is produced. This does not provide evidence of chemical reaction. taking place. So it's false because uh, what determines the chemical reaction to happen is the color change, temperature change, odor change, and the formation of bubbles. Okay, so those are the things you must consider. You must consider for chemical reaction to take place. So this is false. Okay, because color change is one of the indicators that chemical reaction is taking place. Next one, how does an enzyme influence the biological reaction? Letter A, it reduces the speed. That's wrong. It does not reduces. It speed up the rate of reaction. So that's letter B. Okay. Enzymes are catalysts, right? All enzymes are catalysts, and they speed up the rate of the chemical reaction or the reaction. Okay. Next, in humans and other multicellular organisms, which substance is the main energy source? Main energy source of humans and other multicellular organisms are carbohydrates. Okay, they're a source of energy, or the glucose, which is usually converted by the mitochondria in the cells in the process of cellular respiration to produce the energy called the ATP or adenosine triphosphate. Okay, so it's carbohydrates. Next one, amino acids are the building blocks of which macromolecule? Amino acids are building blocks of proteins, so that's letter D. Building blocks of carbohydrates are monosaccharides or simple sugars, which are composed of galactose, fructose, and glucose. Okay. Lipids, the building blocks of lipids are fatty acids and glycerol, wherein the building blocks of nucleic acids are nucleotides. Okay, those are the four macromolecules we have. Next one, an organism's genetic information is stored in which type of macromolecule? Nucleic acid, carbohydrates, lipids, or proteins. So it's in nucleic acids. Okay, nucleic acids. Okay, or the DNA or the genetic information is stored in nucleic acids. Next one, which of the following is not the function of proteins? Builds tissues as bone and muscle, so it is a function of protein. Letter B, transport molecules such as oxygen. It is. Okay, so transport proteins. Next one, control the rate of reactions and regulate cell processes. So it's not the work of proteins. Okay, so this is not the work of proteins. So the answer here is letter C. Okay. Number 4, store and transmit genetic information. This is the primary work of protein. Okay, so the DNA. Next one, 22. Identify the reactants in the chemical reaction. CO2 plus H2O yields H2CO3. Okay, so our reactants here are Our reactants here are CO2 and H2O. Okay, so that's letter B. H2CO3 is the product. Okay, remember that. Okay, so let's go to mitosis and meiosis. Mitosis is a cellular process that produces two genetically identical daughter cells. Okay, because there is no such thing as like synapse. Mitosis happens in the body cells or somatic cells, so it must always be the same. Okay, so the two daughter cells have the same number of chromosomes. This is 46 chromosomes or 23 pairs of chromosomes, and the same as the parent also. Okay, so there you go. Number 2, the first stage of mitosis where chromatin condenses to form chromosomes is blank. It's prophase. Okay, in the prophase stage of mitosis, usually the chromosomes or the chromatin. Okay, chromatin is they are thread-like structures. During prophase, these thread-like structures of chromosomes or the chromatin, they condenses by making a spiral-like structure. Okay, spiral-like. So they form the condensed chromosome. Okay, that happens in the prophase. Interphase, this is the longest part of the cell cycle, where the gap 1 S phase and the synthesis phase and the gap 2 phases happened. Okay, so this is the longest part of the cellular cycle. Okay, prophase is condensing of the chromosomes from thread-like structure to condensed chromosome. Metaphase, this is where the chromosomes line at the center. Okay, so anaphase, this is where the spindle fibers pull the chromosomes and cut it into half. Okay, telophase, this is where the nuclear envelope is what's this nuclear envelope what's that formation. Okay, and then after telophase there is what we call cytokinesis, which is the cell division of the cytoplasm. Okay, so we have here in the next one the part of the cell cycle where a cell spends most of the time carrying a metabolism is interphase. Okay, so this is what I told you in number 2. Next one, the stage of mitosis where the chromosomes line up at the cell's equator. I told you this one also in number 2. This is metaphase. Okay, line up. I'll show you a picture later. There's a question of that. Next one, the stage of the mitosis where the spindle fiber pull the chromatids toward the centrioles. That is in anaphase. Okay, so this is what I'm telling you. The picture here in diagram 1, the stage of mitosis shown in diagram 1, this is metaphase. Metaphase is when the chromosomes, as you can see the dark there, line at the center. That is, those are chromosomes line up at the center, and that area is, or region is, what we call metaphase plate. As you can see there are like fibers that pulls the chromosome towards the two ends of the cell. We call them spindle fibers. And if you see the two holes there, or something like eye-like structures, we call them centrioles. Okay, that makes or produces spindle fibers to pull the chromosomes towards the two ends of the cell. Okay, for the cell division to happen. Okay, so this stage is metaphase. Okay, so the stage of mitosis shown in diagram 2. Okay, so as you can see here, like the chromosomes are now cut into half, and they are pulled by spindle fibers towards the ends, or both ends of the cell. We call this stage is what we call anaphase. Okay, so we have here prophase, metaphase, anaphase, and telophase. Okay, when the nuclear envelope or nucleus are now start to form in after this pulling, that is where telophase is happening. After telophase, this cytoplasm or whole cell will divide into two. That is already cytokinesis. Okay, what is the function of the centromere? Centromere, this is what holds the sister chromatids or chromatids together. Okay, so try to check the chromosome. This is found the circle something at the center of the chromosome. We call them centromere. Okay, so this hold the sister chromatids together. That's letter A. Next one, how many chromosomes do human somatic body cells have? Somatic body cells have 46 chromosomes, or 23 pairs of chromosomes, or we call them, body cells are called diploid. Okay, whereas the gamete cells, or the sex cells or reproductive cells like sperm cell and egg cells, they have haploid, or 23 chromosomes only. Why 23? Because if the sperm has 23 and the egg has 23, when they mate together, they form 46 total number of chromosomes, which is diploid number. Okay, next one, we have how many chromosomes do human gametes? Gametes are eggs and sperm, and reproductive cells or sex cells, they are called they have 23 chromosomes only. Okay, 23 chromosomes, but not 23 pairs. 23 chromosomes, because egg plus sperm, if each of them has 23 chromosomes, so that totals 46, which is the normal number of chromosomes for human cell. Okay, next one, how many gametes form from a parent cell that undergoes meiosis 1 and meiosis 2? There are 4 daughter cells. Okay, so from one parent cell, 4 daughter cells. After meiosis 1, there are 2 daughter cells, and then these 2 daughter cells are going to undergo cellular division again, second stage, and that's meiosis 2, and that forms 2 for each, so 2 times 2, that's 4 daughter cells all in all, so it's letter D. Next one, 13, cytokinesis is the division of the cytoplasm. This is what I told you earlier. Okay, so what factors limit the cell size? What factors limit the cell size? DNA, diffusion, surface area to volume ratio, or all of the above, or B and C only. So it's letter C. Okay, letter C, because the smaller the cell, the effective it works. Okay, the smaller the cell, the more effective it works. The bigger, the weaker. Okay, So that's letter C. Next one, 15. What is gene? A gene is a segment of DNA that controls the production of the protein. Okay. Next one, what is a cancer? Cancer is a malignant growth resulting from uncontrolled cell division. That's, what's that? Cancer. Whereas the karyotype, this is the arrangement of chromosomes, which is number 17. Okay, the arrangement of chromosomes used to detect genetic abnormalities. Next one, gametes have blank the number of chromosomes. We have haploid, okay, or 23 chromosomes, okay, haploid. Diploid is 46 chromosomes or 23 pairs. 23 pair means 2. So 23 times 2, that's 46, okay? Next, a zygote has blank number of chromosomes. A zygote has a diploid number of chromosomes because a zygote is a fertilized egg cell. So when you say fertilized egg cell, there is already one sperm cell inside. So egg cell, it has 23 chromosomes and the sperm cell has 23 chromosomes. So 23 plus 46 chromosomes, which is the normal number of chromosomes for a human cell. Okay, so zygote has already life. Okay, because it's a cell, human cell na siya. Next one, why are there two sets of phases in meiosis? To produce a double number of chromosomes in each gamete or to produce half number of chromosomes in each gamete. So this is letter B because why there are two? Because in the first meiosis produces diploid or 46 chromosomes, but the second meiosis or meiosis 2 produces haploid number of cells. Okay, so that's half. From 46 becomes 23, so that is half, letter B. Okay, so I hope this one helps you a lot, guys, and don't forget to subscribe and hit that bell for notifications of my future videos po. Stay safe, everyone. This is Teacher Seppi. God bless.